everybody, thanks for coming. My name is Angie Reyes and I'm going to share with you the evolution of sexual dimorphism in brain size and brain region volumes across wild guppy populations, developed by the Neurogenetics and Genomics Lab from Los Andes University. The evolutionary explanation for a wide diversity in animals' brain size and neuroanatomy could be linked to the cognitive demands of different animal species. For example, we know that generally observe a trade-off between gut size and the size of the brain because of the cost of building a larger brain. Poecilia reticulata is a small freshwater fish in which we can see a remarkable sexual dimorphism where males exhibit a large variation in coloration. The main sexual traits that guide female preference is based in the coloration in addition with a tail size and the size of the gonopodium, which is the anal fin modified to serve as a copulatory organ. On previous studies, Collin and collaborators selectively bred guppies for large and small brain size relative to body size. They found difference in cognitive abilities between large and small brain lines, as we can see on this graph. Further research on these selection lines showed there is a direct association between brain size and the different sexual traits in males, including tail length, gonopodium length, iridescent area, black area, and orange area in male guppy's body. In order to understand how the brain is evolving in nature and whether there is a relationship between brain size and sexual traits as we saw in laboratory, we set up a study to investigate the variation in brain size and neuroanatomy in the wild, and to investigate the relationship between brain anatomy and sexual traits in nature. In order to complete this study, we focused on an explored guppy system across Colombia. In Colombia, guppies has diversified all over the territory in diversity of environments. We collect a total of 18 populations. We initially went out to the field. For each population, we collect 20 individuals minimum, 10 males and 10 females each. Each individual was measured and photographed to collect data on sexual traits. The sexual traits we measured were tail length, gonopodium length, black and orange area, and orange color saturation in male's body. Finally, we collect data on brain size and anatomy by dissecting the brain. Here we weigh the whole brain, a standard measure of brain size, and measure 5 brain regions: telencephalon, optic tectum, hypothalamus, cerebellum, and medulla. Now, the results. To evaluate difference across populations, we perform analysis of covariance and linear mixed models using the whole body length as a covariate in order to correct for body size. In this graph, we can see remarkable difference in the brain weight of females between populations. It is the same in the case of males. Additionally, we found significant difference between both sexes. This graph shows the mean and the standard error of brain weight variations across populations. In this graph, we can observe variations in the degree of sexual dimorphism between populations for brain weight. In addition, we see that some populations show some drastic, as in this case, difference in relative brain weight for males and females, while others, both sexes, have the same relative brain weight. This difference in the degree of sexual dimorphism in relative brain weight across populations probably reflect difference in the selective pressures imposed by the environment in different populations. This graph shows the mean and the standard error for the proportion of brain regions relative to the whole brain across populations. In case of optic tectum, the graph shows that there is not a notable sexual dimorphism in intervened environment as there is in lotic and lentic. Also, we see that the optic tectum is the only region in which sexual dimorphism favors a larger size in males. In the rest of the structures, we see a larger size in the females. This variation was tested for its brain region with a general mixed model, showed difference across populations between sexes and in the degree of sexual dimorphism across populations, indicating that the brain regions of females and males are responding to different selection pressures. This is consistent with the findings made by Redon and collaborators when they affirm that the brain size of females tends to be maintained between generations in a more conserved way. Instead, the brain of males is more susceptible to predation as a selection pressure. However, we did not find a significant sexual dimorphism for the hypothalamus and cerebellum, but we did find difference in the size of each region volumes across populations. Finally, we performed a linear mixed model with body length as a covariate and population as random effect. We found that orange color saturation is significantly associated with relative size of the various brain regions, and we did not find significant association between sexual traits and female total brain size that explain the variation. Also, in females, we found significant relationship between black color area in male's body and the volume of the optic tectum and the cerebellum, and between the length of the gonopodium and the volume of the medulla. For the males, we found a significant relationship between black area and the volume of the cerebellum, and between the length of the gonopodium and the medulla. In conclusion, our results are consistent with the hypothesis that the proportion of brain regions in females and males responds to different pressures. Furthermore, we see that indeed, when we step out of the controlled environment of a laboratory and face with changing selective pressures, we see that the proportion of brain regions are responding to these pressures differently. I want to thank everyone in my lab and many collaborators that help us with the sample collection. Thanks for coming, and please let me know if you have any question.